Assalamualaikum. My name is Husna Izzati binti Sani. My nomenclature is DN180070. My supervisor is Dr. Nadira Hasraf. So the title of my project is Development of a Bilayer Transdermal Wound Healing Patch Loaded with Bromelain, Kinetic Drug Release and Antibacterial Study. Wound healing is the process comprising of healing of dermal and epidermal tissues by the regeneration. It involves a consecutive cascade of stage of inflammation, migration, proliferation, and maturation. Wound dressing play critical roles in wound treatments. An ideal wound dressing should protect the wound from external contaminants and facilitate the healing process. Electrospin nanofibers show great capability for wound dressing due to their special properties such as high surface area, and as a result, electrospin nanofibers can efficiently absorb exudates and maintain the wound moisture. In addition, bilayer wound dressing, which are composed of two layers with different properties, have gained lots of attention. The difference in the structure and characteristic of each layer can provide several advantages in wound healing application. Next, we go to the statement of the problem. Several current wound dressing have shown some disadvantages that makes the wound healing process ineffectively. For example, hydrogel dressing can hydrate the wounds to provide a moist environment and promote autolysis debridement for wound scab. However, this one-layer wound dressing are unable to meet all clinical requirements due to its characteristic and limitation. Hydrogel dressing is prone to causing skin emission surrounding the wounds, necessitating the use of a second layers of dressing in wound treatments, otherwise it possibly resulting in germs infection and contamination. Next, PVDF has a poor reactive side chain with hydrophobic properties which can promote inflammation in the host cell or tissues and make it less capable of acting as a drug carrier without the aid of bioactive compound. For that reason, some modification and enhancements to those specific properties are theoretically necessary. PVA is a biocompatible polymer that has been widely used in advanced biomedical applications such as wound dressing. However, PVA hydrogels may form undesirable elastic and stiff membrane which restrict their use as wound dressing. Therefore, by combining this material with electrospin, it can solve the limitation of both polymer and produce better polymer composite. Next, the objective of this study first, to develop transdermal bilayer wound healing patch by post-loading of bromelain into electrospin hydrogel scaffold. Second, to characterize the properties of bilayer bromelain scaffold and the drug stability. And the last one is to evaluate the kinetic drug release trend and its effect toward antibacterial activities. So, the scope of this study will be focused on, firstly, PVA PVDF nanofiber mat will fabricate by using electrospinning method. Secondly, PVA collagen HNT hydrogel can be made by using freeze-thaw method. In physical test, will involve scanning electron microscope and atomic force microscopy. In structural and thermal tests will involve Fourier transform infrared spectroscopy, Raman spectroscopy, and X-ray powder diffraction. In kinetic drug release, will use phosphate blocking salt solution in the sample for evaluation. Antibacterial tests will using this diffusion system with E. coli and S. aureus in the sample. Next, we will go to the literature review. Okay, this part is about wound healing process. The wound healing process generally involves four distinct stages, which are hematosis, inflammation, proliferation, and remodeling. Once an injury takes place, the wound healing process starts immediately with the hematosis stage, which involves coagulation to prevent massive blood loss by aggregating platelets. An inflammatory phase, occurring simultaneously with the hematosis, is characterized by the release of several pro-inflammatory cytokines, cytonic peptides, proteins, reactive oxygen species, and growth factors, allowing the wound healing. At the proliferative phase, granulation tissues is produced because of the growth of capillaries and lymphatic vessels from existing vessels present at the sites of injury. Finally, in the remodeling phase, a new tissues is continuously remodeled until its composition and properties are close to those of health healthy tissue. Current treatment in wound healing. There are 4 types of wound dressing, which are passive dressing, interactive dressing, advanced dressing, and bioactive dressing. Hydrogel dressing are made comprised of hydrophilic, inflatable, and insoluble components that come in the form of gel or film. Hydrogel dressing are used to cure dry wounds due to the formation of a damp environment that eliminates dead tissues, and foreign elements from the wound. PVA is one of the most often used polymer in the form of a hydrogel and its great biocompatibility, making it suitable for a variety of biomedical applications, including wound dressing. Polylactides (PLA) is frequently used as the major component of electrospun composites for biomedical application due to their biological properties. Active compound in wound healing. Bromelain as an active compound is the extract obtained from the stem and fruit of the pineapple that contain a high level of proteolytic enzyme. Bromelain has a strong effect on pain mediators such as bradykinin that reduce the bruises and speed up the healing process of wounds. Bromelain also lowers plasma kininogen, which inhibits the formation of kinin that will cause the inflammation, pain, and swelling. Current status engineered scaffold loaded with active compound. Artificial scaffold have been applied and used as a supporting structure for cell cultures and domination of cell growth in the repair or impaired tissues or organs. Bioactive compounds are often used in tissues engineering to promote adhesion, proliferation, and differentiation by facilitating interaction between scaffold and cells. In other research, chitosan scaffold have been proven to be capable of incorporation and releasing active compound as controlled release device. Next, we go to the electrospinning. The formation of nanofibers applying the electrospinning technique relies on the uniaxial stretching of the viscoelastic solution or melt under electrostatic force. The electrospinning process enables the creation of interconnected networks from nanoscale diameter fibers that are similar to the native structure of the natural ECM, promoting the normal function of the cell. Nanofibers can absorb exudates efficiently and provide a moist environment for cells respiration and proliferation due to the physical property. The hydrogel is a type of soft biomaterial that possesses a 3D structure that has a network of the hydrophilic polymer. The hydrogel can be fabricated through the cross-linking process. The insoluble hydrophilic structures have the remarkable ability to absorb wound exudates while allowing oxygen diffusion to accelerate healing. In this part, about the material used in this study. First, PVDF is a piezoelectric polymer which can generate electricity through mechanical stress without the presence of an external power source. PVDF electrospun membrane have a rigidity and low flexibility that cause damage to regenerated tissue interacting with the membrane due of the high hardness and elasticity modulus of PVDF. The material or process combination PVDF and zwitterionic copolymer reported lead to the formation of nanofibrous membrane able to maintain an optimal environment for diabetic wound healing. Introduction of high crystallinity significantly increased the tensile strength of the hydrogel. PVA-based dressing usually contain other materials or pharmaceutical for better antibacterial properties and to speed up wound healing. Collagen have a potential to stop bleeding rapidly and repair a variety of soft and hard tissues. Collagen deposition and remodeling contribute to the enhanced tensile strength of the wound. Collagen-based biomaterials have been used for wound dressing because of their biocompatibility, low immunogenicity, ability to attract wound healing responsive cell, and ease of application. HNT able to act as a reinforcement for the nanocomposite scaffold, which will directly result in the enhancement of the mechanical properties of the nanocomposite scaffold. It was reported that in vivo wound healing evaluation confirms the enhanced healing ability of the chitosan sponge by adding HNT. The last one is the bromelain. Bromelain is considerably absorbable in the body without losing its proteolytic activity and without producing any phagocytes. Previous studies show that this polymer has good biocompatibility and positive effect on wound healing. That's the preparation of Bilayer composite bromelain. Firstly, the preparation of electrospoon PVDF PVA. PVDF will be dissolved in dimethyl sulfoxide solution, then dissolving PVA and mix to form PVDF PVA polymer solution. So the polymer solution continue to electrospinning follow the parameter. To prepare the bilayer composite, the PVA, collagen and HNT are mixed together to form polymer solution. Then the solution will pour onto PVDF PVA electrospoon in mold by using freeze-thaw method to form bilayer composite. Lastly, bromelain will dissolve with distilled water in different concentration. Then the bilayer composite will loaded with bromelain using post-loading method to form bilayer composite bromelain. The properties of scaffold can be characterized with testing method including SEM, AFM, FTIR, XRD and Raman spectroscopy. For ATM, it will provide extremely high resolution image of the surface topography sample structure. For SEM, to determine the mean diameter of the sample. For FTIR, determination of chemical characteristic of sample. For Raman spectroscopy, to study the bonding behavior and phase purity of sample. And the last one, XRD, to evaluate the crystalline of beta phase of sample. In kinetic drug release, after Bilayer composite sample will be loaded with benzyl colonium chloride and dried the sample. The dried sample immersed into phosphate buffer saline, and the absorbance of the sample was observed to evaluate the drug release pattern. For antibacterial test, the bilayer bromelain scaffold will each placed onto a long S. aureus and E. coli on the agar plates and incubated overnight. The zone of inhibition formed around the scaffold will be observed against the two microorganisms. In the expected result, the incorporation of bromelain with the electrospoon hydrogel scaffold will enhance the process of healing. The properties of bilayer bromelain scaffold enable to characterize by using SEM, AFM, AR3D, FTIR, and Raman spectroscopy. This analysis will confirm the existence of bromelain in bilayer composite. The kinetic release of bromelain will be expected to follow the friction diffusion model by minimizing the burst area effect in the initial stage of implementation. The bilayer strategy will help to prolong kinetic release by altering the diffusion of fluid during the real application. The antibacterial efficacy will be determined by the inhibition zone, which is the area surrounding the disc where the bacteria have not grown large enough to be visible. The vast availability of bilayer transdermal wound healing patch loaded with bromelain will produce a sustainable wound dressing in the future. So in the conclusion, first the overview of wound healing electrospoon and polymeric nanofiber and hydrogel as a wound dressing. And then focus on assessing the kinetic drug release trend and effect towards antibacterial activities of bilayer composite bromelain scaffold. Next, the use of electrospoon layer show good absorbing properties of exuded, and then develop in bilayer structure by layering PVDF PVA electrospoon with PVA collagen HNT hydrogel to enhance the wound healing process. Release profile and low cytotoxicity that will have become the effectiveness of bromelain in wound treatment, and also exhibit antibacterial properties in preventing microbial proliferation around the wound area. This project also be an advanced dressing for wound healing application in tissue engineering due to its excellent properties. This research hope will give knowledge contribution in biomedical industry field, especially towards on development of bilayer transdermal wound healing patch loaded with bromelain. That's all from me. Thank you.